Post and Rudolph 2018. So we called about the picocolites. Picocolites have a lot of importance in various fields. In majorly for use of agar, alginate, alginic acid, and carrageenan. So carrageenan is a linear sulfated polysaccharide commonly classified into three types, that is kappa carrageenan, iodocarrageenan, and lambda carrageenan. Based on that sulfated group present in the sulfated groups, kappa carrageenan have a one sulfated group, produces strong gel with the potassium and calcium salts. Iodocarrageenan forms a elastic gel with the calcium salts, and lambda carrageenan having three sulfated groups does not produce strong cell because it does not have a free hydroxyl group, so it only forms a viscous solution. So these are the three kappa and iodo or lambda carrageenans have the major value or major importance in food, beverages, and pharmaceutical industries. Nowadays its demand is increased over 5 to 6% every year. So these are the seaweed diversity of the Indian coast. So overall, in India, we have 434 species of rhodophyta and 191 species of phaeophyta and 3 species of xanthophyta, that is yellow green algae, and 216 species of chlorophyta. Totally 844 species seaweed diversity we have in India. In percentage, in red algae genera, we have red 51% and brown 23% and green 26%. From red algae in 51%, we have 12 genus of carrageenophytes, that is carrageenophytes available for carrageenan production in India. That 12 genus we have, among 12 genus we have 66 species of carrageenophytes, that is seaweeds available for carrageenan, including for kappa carrageenan source, lambda and iodocarrageenan source are mixed carrageenan source. But in India, Hypnea and Kappa bicus alvarezi is the major source for carrageenan production. In worldwide, Chondroscripsis gigatina species, Kappa bicus alvarezi is the major source for carrageenan in various countries. These are the carrageenan production value reported by FAO report 2022. These Euchemia and Kappa bicus alvarezi is the dominant species to dominate the whole carrageenan production through this entire world. For the percentage of 23.2 for Euchemia species and Kappa bicus alvarezi 4.6 and Euchemia denticulum 0.4%. These carrageenan production values involves in the global carrageenan market size valued at $780.5 million USD dollars. It increased every year for especially for carrageenan market for 6% in 2022 to 28. These are the carrageenan market report scope. For market size value in 825 million score, revenue forecast expected in USD dollar is 1,248.6 million. Revenue growth rate was expected for 6%. For in kilotons for produced in the fresh or wet white, the growth rate was expected for 3.4% from 2020 to 2028. Market demand increased 54 to 68.45 kilotons. Country scope including India and various countries. These are the production of Kappa bicus alvarezi cultivation. Why I am saying Kappa bicus alvarezi means this is the major source of carrageenan in India. The Kappa bicus alvarezi cultivation is involved in the red seaweed cultivation overly. From 2000 to 2010, it produced 5.6 million fresh white, correspond to an over 63% of red seaweed cultivation. And the commercial production is reached 7,187 dry mass worth rupees 140 million farm gate. The annual production trend was found be the 69.37% from farm gate price, and it gives the production for 236.05 average value fresh weight value and average growth rate of 5.26. Why 5.2 means when the seaweeds or other plants gives the cultivation growth rate above 3% means is suitable for commercial cultivation. These are the graph detailed about the 2000 to 2010 growth rate for Kappa-Picus alvarezi. These are the Kappa-Picus alvarezi cultivated by different methods, that is raft, monoline, tube net method. Even tried net bag and other also method, but successfully suitable for commercial cultivation is raft, monoline and tubular net method is chosen. These are the multiple value ordered products from integrated process from Kappa-Picus alvarezi plants. These are the schematic diagram from various products derived from Kappa-Picus alvarezi, such as biostimulant, biofilms, biofuels, low sodium salt, etc. etc. Biostimulant, it's a plant biostimulant. This is derived from the carrageenan rich seaweed extract. And this sap contains micronutrients to help to encourage the growth of the plant. The estimated concentration for axin, cytogunin and gibberellins are given and present in the seaweed sap, that is 56.04 for axin, 77.20 for cytokinin and 17.38 and 128 for cytoskinin and gibberellin respectively. Compared to chemical fertilizers, this is derived from seaweeds or biodegradable, non-toxic and non-polluting and non-hazardous to human, animals and bats. And one more thing, the plant biostimulant based on CSM technology have a patent right and to marketed a plant-based biostimulant Saccharika for marketed to the Indian Farmers Fertilizer Cooperative Limited will be soon as the name of Saccharika is a plant biostimulant. These are the graph detailed about the private sector. Agri produced the Kappa-Picus alvarezi sap throughout the year 2018 to 2015. Other value added products, that is mainly in biofuels. Among tested 25 algae, 23 algae gives satisfied results. That is highest value of Euchemia carni 40 to 4%, in biohydrogen Zelidium 1C, and biobutanol laminaria species and biomethane also laminaria species give the highest yield in biofuel production. Summary, and not only the carrageenan not only involved in biofuel production and biostimulant, also it involved in animal feed and confectionaries industries. Carrageenan is a sulfated polysaccharide which is produced from the red algal genera only. Annually consume over 232,000 carrageenophytes to produce 57,500 of carrageenan, having a value over of $518 worldwide. In India, the world carrageenan production strongly dependent on Euchemia and Kappa-Picus alvarezi. In India, only Hypnea and Kappa-Picus alvarezi have the cultivation technology to produce the carrageenan production and are major source for the carrageenan production. Carrageenan also approved as a thickening and stabilizing agent for by FDA in USA. And other than for drug committees and national industries also allowed to use a particular recommended amount to use as a stabilizing and thickening agent. Apart from this carrageenan, it used as a biostimulant, bioethanol, cellulose, biodegradable polyethylene bags and confectionaries and various fields like cosmetics, nutraceuticals and various fields we are used directly carrageenan or indirectly in other seaweed sources. Thank you. Now I express my sincere thanks to DST SERB and CSA New Delhi for financial support for this work. Thank you.
Thank you. Very good presentation. Any questions? Please ask.
Firstly, they are the floating bamboo raft method. Second, only it depends upon
Sir, one question or basic question. அதாவது இந்த இந்தியாவில் ப்ரொடக்ஷன் கம்மியாகுங்கிறீங்க இல்லையா, கம்பேர்ட் டு அதர் கண்ட்ரிஸ். ஸோ ஃபார் எக்ஸாம்பிள் இப்போ ஜப்பானில் வந்து ப்ரொடக்ஷன் கூடுதலாக இருக்குங்க, பட் அந்த சீ ஷோர் வந்து கம்மியாக தானே இருக்குது கம்பேர்ட் டு இந்தியா இல்லையா?
அவங்க லேண்ட் பேஸ்ட் கல்ச்சரும் they are using sir. Aquaculture in IMS aquaculture integrated aquaculture also they are using. But and one more thing they are using daily custom manner they use
இல்லை அதுக்கு
food grade algae. So that they give importance to food.
அது கேட்கல. ஆக்சுவலாக இது நமக்கு இவ்வளோ கோஸ்டல் ஏரியாவெலாம் இருக்குதுன்னு சொல்கிறீங்க இல்லையா, பட் என்ன ப்ராப்ளம் இதில் இருக்குது, அது லார்ஜ் ஸ்கேல் ப்ரொடக்ஷன் இல்லாமல் இருக்கிறதுக்கு அப்படின்னு? இல்லையா? ஸோ அது ஏதாவது கிராப்பில் வந்து ஈல்டு எத்தனை பர்சன்டேஜ் கூட்டிருக்கு அந்த மாதிரி டேட்டாலாம் இருக்கு இல்லையா? சரி, எந்த எந்த கிராப்பில் வந்து ரொம்ப கூடுதலாக காட்டுது? நம்ம கேரளாவிலலாம் கேரளாவில் நாங்கள் இருக்கும்போது இந்த இதெல்லாம் யூஸ் பண்ணியிருக்கிறாங்க. ஆக்சுவலாக நம்ம சொல்கிறது இந்த கமர்ஷியல் கிராப்னு சொல்கிறாங்க இல்லையா, பிளான்டேஷன் கிராப்ஸ் அப்படிங்க. இந்த காடமெல்லாம் நாங்கள் யூஸ் பண்ணி பார்த்து டேட்டாலாம் எடுத்துருக்கேன். ஸோ அதனால தான் நான் கேட்குறேன். அது வந்து சாதாரண பிளாக் கிராம் அதெல்லாம் வந்து சாதாரண கிராப்னுங்கிறாங்க இல்லையா, கேஷ் கிராப்பில் வந்து யூஸ் பண்ணுறது வந்து நாங்கள் ஒரு ப்ராஜெக்டை ரன் பண்ணி டேட்டா
So are you giving chance to the local farmers? How to cultivate this kappa bagas? Are you practicing? Yes, the local farmers.
Yes, yearly we conduct a program to produce a seedling material for the farmers for free. So we have a project PMI scheme. So every year in a 3 months basis we given a lot of seedling material for seaweed farmers around from Rameshwaram to Tondi. 45 days. Yes,
Your presentation is very good, but nice presentation. Congrats.
Next presentation is assessment of growth and growth, carcinogen, yield and economic feasibility of cultivated kappa physis alvaricis dot using three different. So, C. Perisami from Assistant Professor and Head Department of Botany, Pasuman Muthramalingam College, Melanedunallu, Tenkasi.
Yeah, very good evening to all. Now, a lot of talk after lunch about kappa ficus. Now, my session is also on kappa ficus. So, this is two part. First part, I have worked with earlier CSMCRI, then Aquagri. So, my first part is with Aquagri. What I have done, it's a piece of work. So, apart from my several patents, right? First, kappa ficus alvarici, and second one is self-life. When I was coming out from cultivation, now I am working in Pasuman Muthramalingam College. So in my lab, I am doing some work on self-life of carrageenan. So self-life of that seaweed, what I have collected in 2014. So first introduction, kappa ficus alvarici is an red algae. It's a rhodophysia member. You can see in 1960, Doty and Alvars, they have started cultivating this kappa ficus in Philippines and Indonesia. So it is introduced in 26 countries. Nearly the production already last presenter have mentioned in a current scenario. So kappa ficus production in 2010, first time we have entered in the world market in 2010 about kappa ficus. So in India, as far as India is concerned, in 1995, CSMCRI scientist O.P. Mayer, he is the father of kappa ficus alvarici in Indian waters. So he brought nearly 2 grams. You can imagine 2 grams of kappa ficus with 10 centimeter length. You can imagine how it was grown within a period of 22 years. So Indonesia 61%, Philippines CSMCRI, then transferred in Oka coast. Just what he done in earlier days, because he brought only 2 centimeter. So he cultured in lab. He speaks with seaweed. You can imagine that much scientist is dedicated only for kappa ficus. When I visited Bhavanagar during my PhD work, just he sit with test tube in with kappa ficus, 2 centimeter kappa ficus. He speak with kappa ficus. When you will grow, when you will grow, because he is kept for more than 90 days with only 2 centimeter. So then he standardized the method in lab culture. Then Provosali medium, he cultured with Provosali medium. Then he transferred to Oka coast. Oka coast is somewhat rough, like our open sea. So he just put it in the soil, inside the soil. Then he analyzed for more than 2 months. Then he transferred 1 gram of kappa ficus to Mandamam, Dr. Easwaran. He handed over 1 gram of kappa ficus to Dr. Easwaran. Then he initially started with bag culture, plastic bag culture. Then it is propagated. Then it is transferred to net bag culture. Then technology was transferred to PepsiCo India Holdings Limited in 2000. So once it is technology transferred to PepsiCo, then they go for mass cultivation, commercial cultivation. Initially, they have started with Kudumba model cultivation. In Tamil, they want to started with Kudumba model cultivation. Up to 2004, they have done with only monoline culture, with net bag or sometime bag culture or sometime open culture. So in 2005, problem started because it is a MNC. So people are sticking on MNC. So some people are objecting. Then when I was with CSMCRI, we have done cultivation in Crusade Island, right? Crusade Island, south of Crusade Island. It looks like a lagoon. Then reef is there, that is Galaxura reef. So cultivation is going on, then controversy started. Once the earlier that islands are under the guidance of fisheries. Once it is come to forest, they asked CSMCRI to evacuate in one fine morning. So the culture, whatever they have cultivated in Kappa Vegas is kept as such in the sea. So they are evacuated and come to mainland. So those seaweeds, it is growing only in vegetative. If the temperature is less than 2, it start producing spores. So the problem we will discuss on tomorrow. So then technology was transferred to PepsiCo. Then this cultivation is going on in all the coast. Even in Tuticorin, we have started in 2004. First, we have started in Tharuvikulam. Then we have started in Hare Island and some places in there. So now I go with my work. So to study the feasibility and economic of Kappa Vegas, we have started with 3 cultivation. So people familiar with different cultivation. So nearly 18 cultivation model is there. If you think some cultivation, you can patent it. So that much work is there. So if you work seriously for at least 1 year, you can patent one technology. So it is a very secret. You can try with some technologies. So 3 method we have started. Floating bamboo raft method, off bottom monoline net fenced method. Third one is off bottom monoline net fenced method. This is for people, right? Then one company want to study the carrageenan self life, that is Aqua Agri and PSS Ganesan and Sons in Tuticorin. They want assign me to find the self life of that seaweed. How many periods we can store the seaweed? If India now we are in a problematic condition for Kappa Vegas production, but they are thinking that if Kappa Vegas is produced in large scale, whether we can store for few years. So for that study I have done in second one, that is in my lab. So this is the cultivation place at Vedalai near Rameshwaram, before Mandapam. It is there. So I have pointed out that. Then this is the raft method, right? This is the raft method. It's a very simple model. 45 rafts will be there. One person can handle 45 rafts. Every day he has to harvest one raft and he has to replant another raft, same raft on the same day. So they will get the continuous income. The income is very lucrative income. They can get minimum 20,000 per month. So this is the second model. We have tried with net fence, right? Net fencing for, this is for 90 lines, right? 90 rafts we have maintained, then 90 lines we have maintained by one SHG, self-help group, Sri Vigneshwar Adavar SHG in Vedalai. So then third model is off bottom monoline netback method. So during northeast monsoon, people will maintain this netback method. So we have tried with these 3 methods. Then afterwards, after 45 days, every 45 days, we have collected the monthly data. Then for from the cultivated material, harvested material, we have just dried in sunlight. Then we have do for SRC using Stanley and Ono method. Then we have find gel strength also by Bloom method to whether our quality is good or not. As far as India seaweed is concerned, this Kappa Vegas is concerned, we are very superior with even the native place of Philippines and Indonesia. We are having a superior quality of SRC than any other country, right? Mars, MERS, they are the leader producer in the world. They have visited our farms and they have purchased seaweeds. They have certified that our Kappa Vegas dry weed is superior than any other country. Then we have analyzed the environmental factors, temperature, salinity, nutrients. Then occasionally in monoline culture, if it is in open, there will be a grazing. So for the Crusader Island also, that is the major problem. So this plant is trapped in a coral, in Acropora coral, it's a finger corals, just trapped a bit when it was evacuated. So that bit is grown. The same time grazing will also happen. This plant is growing very fast. 45 days it goes to even the period already they have mentioned. So DGR is 3%. You can imagine every day it will grow 3%. So growth will be more. So grazing will be there. If it is open, it will be grazed by the grazers, mainly by the fishes. Then epiphytes will also be there. Then I have calculated the economics. So now we will see the what are the results seasonal using 3 different methods. You can, I have mentioned the mean annual 254.76 per raft after 45 days. Then as far as net fenced method is concerned, 280.13 for per 60 meters. Raft is 60 meter. Same way, the monoline in net fenced model is also 60 meter. Then net back, right? 150 net backs in 60 meters. That gives 266.12 kg. Initial weight is 60 kg. After 45 days, you will get 254 kg, 280 kg, 266 kg. Then from this matured plant, just you can harvest 60 kg and replant the same raft on the same day. The balance material will be dried for sale. Second one, daily growth rate. With the growth formula is there, we have find daily growth rate. If the daily growth rate is more than 2.5, it is viable for commercial cultivation. We have got 3.2, 3.4 and 3.28 for these three methods. Then as far as carrageenan yield is concerned, if commercial carrageenan is more than 25%, it will be viable for commercial carrageenan production. So we have got more than 30% in all the methods. Then temperature I have mentioned. So average of this, as far as kappa biogas is concerned, it required 30 plus or minus 3%, 3 degrees C is good for cultivation. Then salinity we have find. Then nitrate and phosphate. Major problem is if nitrogen and phosphate is there, the plant will grow very well. Then shelf life. So we have submitted the result also to that company. So as far as shelf life is concerned, the shelf life is more in open monoline net fenced method because it is open. So it faces a lot of any problem arises, it faces directly. If it is in net bottom, it is in peripheral. If it is in net back, it is, you can imagine people, children, for example, if you take children, if they are growing in open environment, they are having lot of immunity. If it is growing only in AC, then if they are going out, then they will face lot of problems. Same way, this plant, if it is grown in open, it has a good carrageenan yield, good growth also will be high because it is exposed to open seawater. So it is more in, after 6 years, there will be a decrease in carrageenan because it is, carrageenan is hot water soluble, not cold water soluble. If it is stored after 6 years, then some degradation will be there, some bacteria is involved, that work is going on. After 6 years, it will create problem. So we can go up to 6 years for this cultivation. Then this is the economics. So as far as raft is concerned, the 1,500 rupees is investment per raft. As far as open monoline is concerned, there will be only one mesh around the periphery. So it cost around 1,000. Then as far as net back is concerned, it cost 2,000. You can imagine for raft we are giving bottom net fencing. The main cost is net. So every raft have a below the bamboo there will be a net. As far as net fencing is concerned, peripheral netting is there. As far as net back is concerned, each plant is protected with net. So the cost will be more in the net fencing method. Then we have calculated with the mean crop yield and all. You can imagine the potential. Potential means because we have take extra care. For research purpose, we have take extra care. If some plant are missing, we have replanted up to 15 days. Up to 15 days we are replanting. Due to heavy wind or some boat movement, plant may fall on the ground. So we have replaced the seed up to 15 days. So this is the potential monthly income. At least the people will get not less than 20,000 rupees per month. This is monthly calculation. As far as floating bamboo raft method is concerned, it the people will get around 25,000. As far as open monoline net fence method is concerned, they will get 28,000. As far as open monoline net back culture is concerned, they will get 26,000. So this is the economic analysis. So with the annual biomass. So last part, because they have given only 7 minutes, so I am rushing up. So final discussion. This method is advocated to economic feasibility of cultivating Coprophagus. It's a very good alternative option to the coastal fisher hawk. Then among these three methods, open monoline net fenced method is superior than all the methods, both daily growth rate, total annual biomass and SRC yield. As far as the industry is concerned, they are getting more because they want more carrageenan. So they will get more carrageenan in open monoline net fenced method, and the shelf life is up to 5 years. There will not be a problem. Next, finally, I want to acknowledge my home institute, Central Salt and Marine Chemical Research Institute, CSMCRI Bhavnagar. When I was after finishing my MPhil, I have joined in CSMCRI. Then this whole work was done by Vigneshwar Self-Help Group in Vedalai. Then Aqua Agri Processing Private Limited, they have helped a lot. Now the officials and officers of Vasavan Muthuramalingetta our college, where I have worked now. Then my guru, he is working with Aquaculture Foundation of India, Madurai. Thank you, one and all. Any questions you can ask?
So now open for discussion.
As far as Coprophagus is concerned, we are facing, as far as government is concerned, earlier up to when it is growing good, government object. Now the plant is not growing well, government is giving 750 crores, not single rupee, 750 crores is kept idle. Okay, as far as cultivation is concerned, people take care, they will get more money. If you think in 2008-9, we have worked in even in Tuticorin coast at near Hare Island, then towards boat. Actually, the government give permission to the north of Bag Bay and south of Tuticorin post. This Gulf of Mannar reserve is kept for idle. They won't allow any people to cultivate Coprophagus. There is a GO in 2003. They have released a GO and by mentioning these areas. So, but nutrient rich is in the restricted area. When we were growing in this Crusade Island, the growth rate was 12%, not 3%, 12%. For cultivation, currently the seed is not viable because in Philippines and Indonesia, they are facing same problem, but the plant is native and they produce spores. By spores, it is viable, but we are not having that much condition because it requires 2 degrees C. Less than 2 degrees C, it produces spores, but we have never faced, we are hottest. So that is the major issue. Yes, yes. Currently it is there due to the non-viable of current seedling. Now CSMCRI has, I think, they will develop by tissue culture by doing some magic, and they will release at least they have give some opportunities to the coastal people. Yes, because we are unable to do. Already the technology was done by CRK Reddy, then Raja Krishna Kumar. So they are having the techniques because tissue culture is not very easy for this species. So it requires sophisticated lab. They are having well equipped lab in Bhavnagar. Open area. Here also they are getting very good income up to 2014. 2014 and 15 they are getting nearly 20 to 22,000. Some people they have purchased a very good bike. Then they have started in Tsunami Colony. They have built big houses. but now they are facing big problem with the non-viable of this seed. Any questions? Government is expecting to import some good viable seeds. Already project we have submitted on the behalf of CSMCRI, then CMFRI. I think some good species may come into India, but exotic algae. It will take 4 to 5 years quarantine, then it will released. Yes, any questions? Yes, ma'am. Ma'am, as far as researches is concerned, ma'am, only 5 to 6 species, promising species are there, but we are not having any good technology. CSMCRA has developed some technologies, but as far as commercial, once it is commercialized, then will be the success. Actually, government is willing to give any kind of fund. As far as trade is also concerned, they will release not 100%. 50% they will release, 50% we have to put. Like that, if government is willing to invest any quantity, but research method, perfect method is not available, madam. Each species have different method. In temperate region, they are doing in pond cultures, pond culture. So in India, prawn culture is not going well. Now they are not allowing Kappa vegas to do. That is the problem. Any questions? Madam, temperature, salinity, temperature or salinity, then